Cellular respiration. Cellular respiration is the most efficient catabolic pathway used by organisms to harvest the energy stored in glucose. Whereas glycolysis yields only 2 ATP per molecule of glucose, cellular respiration can yield 36 to 38 ATP. Cellular respiration is an aerobic process. Oxygen acts as the final acceptor of electrons that are passed from carrier to carrier during the final stages of glucose oxidation. The metabolic reactions of cell respiration occur in the eukaryotic mitochondrion and are catalyzed by reaction-specific enzymes. Cellular respiration can be divided into 3 stages: pyruvate decarboxylation, the citric cycle, and the electron transport chain. 1. Pyruvate decarboxylation. The pyruvate formed during glycolysis is transported from the cytoplasm into the mitochondrial matrix, where it is decarboxylated, as in it loses a carbon dioxide, and the acetyl group that remains is transferred to coenzyme A to form acetyl-CoA. In the process, NAD+ is reduced to NADH. Citric acid cycle. The citric acid cycle, also known as the Krebs cycle, the cycle begins when the 2 carbon acyl groups from acetyl-CoA combine with the oxaloacetate, a 4 carbon molecule, to form the 6 carbon citrate. Through a complicated series of reactions, 2 CO2 are released and oxaloacetate is regenerated for use in another turn of the cycle. For each turn of the citric acid cycle, 1 ATP is produced by the substrate level phosphorylation via a GTP intermediate. In addition, electrons are transferred to NAD and FAD, generating NADH and FADH2 respectively. These coenzymes then transport the electrons to the electron transport chain, where more ATP is produced via oxidative phosphorylation. Studying the cycle, we can do some bookkeeping. Keep in mind that for each molecule of glucose, 2 pyruvate are decarboxylated and channeled into the citric acid cycle. Electron transport chain. The electron transport chain is a complex carrier mechanism located on the inside of the inner mitochondrial membrane. During oxidative phosphorylation, ATP is produced when high-energy potential electrons are transferred from NADH and FADH2 to oxygen by a series of carrier molecules located in the inner mitochondrial membrane. As the electrons are transferred from carrier to carrier, free energy is released, which is then used to form ATP. Most of the molecules of the ATC are cytochromes, electron carriers that resemble hemoglobin in the structure of their active site. The functional unit contains a central iron atom which is capable of undergoing a reversible redox reaction. That is, it can alternatively reduce and be oxidized. Sequential redox reactions continue to occur as the electrons are transferred from one carrier to the next. Each carrier is reduced as it accepts an electron and then oxidized when it passes on to the next carrier. The last carrier of the ETC passes its electron to the final electron acceptor, oxygen. In addition to the electrons, oxygen picks up a pair of hydrogen ions from the surrounding medium, forming water. Total energy production. To calculate the net amount of ATP produced per molecule of glucose, we need to tally the number of ATP produced by substrate level phosphorylation and the number of ATP produced by oxidative phosphorylation. 1. Substrate level phosphorylation. The degradation of one glucose molecule yields a net of 2 ATP from glycolysis and 1 ATP each for one turn of the citric acid cycle. Thus, a total of 4 ATP are produced by substrate level phosphorylation. 2. Oxidative phosphorylation. 2 pyruvate decarboxylations yield 1 NADH for a total of 2 NADH. Each turn of the citric acid cycle yields 3 NADH and 1 FADH2 for a total of 6 NADH and 2 FADH2 per glucose molecule. Each FADH2 generates 2 ATP as previously discussed. Each NADH generates 3 ATP, except for the 2 NADH that were discussed that were reduced during glycolysis. These NADH cannot cross the inner mitochondrial membrane and must transfer their electrons to an intermediate carrier molecule, which delivers the electrons to the second carrier protein complex, Q. Therefore, these NADH generate only 2 ATP per glucose, so the 2 NADH of glycolysis yield 4 ATP, and the other 8 NADH yield 24 ATP, and the 2 FADH2 produce 4 ATP for a total of 32 ATP by oxidative phosphorylation. The total amount of ATP produced during eukaryotic glucose catabolism is therefore 4 via substrate level phosphorylation plus 32 via oxidative phosphorylation for a total of 36 ATP.